Hello friends, welcome to SR Academy.
Welcome to SR Academy. Empowering minds, igniting curiosity. Your gateway to knowledge unleashed. Let's get started.
Today we will discuss about the cell motility and migration in this video. So cell motility and migration, as the name itself indicates, it's about the differentiation and it's about the migration, that means movement. So cell migration that can be assessed by various techniques, such as we are having time-lapse microscopy. Usually the very conventional methods it falls where one can use the microscopy, and in order to identify how the cells are differentiating over a period of time. Imagine I am in the laboratory, okay, you are in the laboratory. Now you want to differentiate the cells. You will initially put the cells and growth factors, scaffolds, everything. Now under the microscope you will see the particular, you will take one photo at or image, you can capture one image at 0 hour. Next, after 24 hours, after 48 hours, next 72 hours, like that, at particular levels or different intervals. For example, after 5 hours, after 10 hours, depending on the cells. If the cells are differentiating faster, then you can reduce the time. If not, then you can increase the time. The broad range you can put from 0 to 1 day, 2 day, 3 day, 4 day, 5 day. If it is required 0 to 1 week, 2 weeks, 3 weeks, like that. So you can have it's always good to have the more amount of data. So you can always go for 4 hours, 6 hours, 10 hours, like that, 2, 3, 4, 2, 4, 5, 2, 4, 6, 8 and all. Usually the people will go. So conventional methods, the time-lapse microscopy was very useful, so that we were able to identify, take the image, then go for this particular colony counter, cell counter, check the hemocytometer, check the how many number of cells are there in every one box, and then multiply with the number of the box which is covering, and that's how we will be able to count the number of particular and we can say that the cell is migrating, that means, or you can say cell is differentiating and it is moving further. So we are having that was about time-lapse microscopy. Then we are having Boyden filter transmigration assay, Zygmunt chamber, Dunn chemotaxis chamber, and also we are having the agarose gel assays. These all are the things which we can use where to check whether the cell is migrating or not. So these techniques allow the tracking individual cells at a specific time. That means at different interval, how exactly the individual cells are there, how many number of cells are there at 0 hour, next after next intervals, like 4 hour, 2 hours, 4 hours, or day, 1 day, 2 day, like that. So I told you, I just gave an introduction about the time-lapse microscopy, how it works. Now next thing is what? Boyden filter transmigration assay. So Boyden filter transmigration assay is nothing but here we will be using transmigration. So there is a filter or a membrane, so which will act as through where the migration will occur. So here Boyden filter apparatus is comprised of the porous membrane, which separates two components or two compartments placed one over another. So this is how it looks like. This one, this is your Boyden's chamber. We are having this. We are having the plate well membrane here. This is a membrane, and these are the cells. Okay, this is the one side view. Okay, so here Boyden filter apparatus comprises of porous membrane, that membrane we saw, which separates two compartments placed one over the other, right? So this is the membrane. Okay. Next, cells are placed in the upper compartment, and the lower compartment contains the chemoattractants, which diffuses through the porous membrane to create the concentration gradient. That means the cells will be initially put here, okay, and here chemoattractants will be there. Okay, so that can be yellowish part, can be chemoattractants. So whenever you put this particular thing, what happens, since the concentration of the cells are high on other side of the membrane, and the different side the concentration of cells are less, so the cells will the concentration gradient will start. What is concentration gradient? When there is a concentration difference, higher to lower concentration or lower to higher concentration, like so. Because of the concentration gradient is formed, diffusion will be there. So cell will diffuse all through this particular membrane and come and settle down here. Understanding? We can see the cells, okay, just below the membrane initially, which was put above the membrane. Understanding? Right? So cells are placed in the upper compartment, and the lower compartment contains the chemoattractants, which diffuses through the porous membranes to create a concentration gradient. Now the cell migrates in response to the chemoattractants via pores. Obviously, when we are talking about membrane, it will have pores. That's why we are calling it as a membrane. Through the membranes, it will be migrating. So now the assay is stopped at specific time points, or for example at specific points, 0 hour, 2 hours, 4 hours, like that. Similarly, and the cells over the membrane, that means just above the membranes, are counted after the fixation and staining. So we will come to know that how many cells have migrated and how many cells have not migrated. Understanding? So next we are having a Zygmunt chamber. Zygmunt chamber that will help to overcome the demerits of Boyden filter assays. So whatever the demerits of Boyden filter assays are there, so those can be overcome by the Zygmunt chamber. So it has a plexiglass side. So the particular Zygmunt chamber has got a plexiglass slide. So these are the plexiglass slides are there. Cover slip is there, wells will be formed, okay, and one bridge will be there. So two wells will be there, and which is separated by the bridge, and the clips are there where the cells can be used. So Zygmunt chamber helps to overcome the demerits of the Boyden filter assay. So it has a plexiglass side which has two rectangular chambers that are separated by a small bridge that is 1 millimeter wide. Okay. So that we just saw in the diagram. So one well is filled with the medium, that is the cell. Another well will be filled with the chemoattractants. So and concentration gradient is formed. That means the concentration gradient will be formed as the chemoattractant diffuse through the bridge. Okay, because higher concentration to lower concentration. Chemoattractants will move from one side to another. Cells will move from one side to another because of the concentration gradient. So now the cells are allowed to grow. That means the cells will be allowed to grow on a cover slip. So this is the cover slip. Okay, cells will be allowed to grow here in cover slip, and once attached, so the slip is inverted and placed over the chamber. That means you can invert after growing here, no, cover slip, you can invert the slip and attach that. The cell will be attached to the particular slips or clips. Okay, slip is inverted and placed over the chamber. Now 3 to 10 micron gap exists between the surface of the bridge and the cover slip, obviously. So from surface to the cover slip there will be around 3 to 10 micron gap will be there. On observing the chamber under microscope, migration can be visualized. So since there is a gap is there, so that you can visualize the under optical microscope, and we can come to know that how many the cells, whether the cells are migrated or not. Over a period of time we can check. That was about the second one, that was Zygmunt chamber. Next we are having the Dunn chemotaxis chamber. So this is nothing but a modified version of the Zygmunt chamber only. That's why I have given here only. So in which the concentration gradient is formed between two concentric wells. So here it is two normal wells which is separated by a bridge. Here it will be centric wells. So two different wells will be created. Okay, depth will be around 0.02 millimeter. Okay, Dunn Weber, England. So that is what we call it as Dunn chemotaxis chamber. Here there also that because of the concentration gradient only, the cells will move in between whatever the towards center. That's why it works based on the centric wells. Okay, so that was about 3 chambers, or we can say Boyden's filter, Zygmunt chamber, and Dunn chamber, which is a modified version of Zygmunt chamber. And these are the diagrams. And Agarow's assay. Agarow's assay simply agar gel electrophoresis, what you use. The same thing. So I have not mentioned any images and all, given images and all, because every biotechnology student, since you are in 7th semester, you will be knowing how does the Agarow's assay works. So under Agarow's assay, it is designed to be simpler compared to the other methods. Definitely, the other methods require significantly designed chamber or specifically designed chamber for performing experiments, whereas agarose assay is one common thing and which will be used for many applications. But this assay utilizes a simple chamber into which the agarose, that is agarose, is poured, make a gel, and two wells will be formed, and that can be purged, or you can just carve it, so you can make two different wells and load one well with the media, another well with the chemoattractants. Now the chemoattractants creates a concentration gradient that and attracts the cell to migrate towards it from the other well, so that the cell migration can be seen under gel electrophoresis. Now we know what is gel electrophoresis. I will just give you the brief introduction. So gel electrophoresis is nothing but an apparatus where you will be using agarose for gel formation, and you will be making holes, so that those are called as wells. So you will be adding a cell in one particular any kind of materials you will be adding in agarose gel electrophoresis. And when you give the power supply, we are having cathode and anode also. So based on that also, what happens, based on the electric thing, if you want to go, you can just go through the we can give the electric potential, so that the migration will be there. And here because of the concentration gradient also, the migration will be there. So here we will be using agarose assay for the with the help of concentration gradient, where the one side you can put the cell media, make one well, put a cells, whereas another side you can put chemoattractants, and chemoattractants will attract the cells, so the cells will start migrating. So you can see that, and that migrations can be visualized in UV luminescence, these things and all, right. The next topic we are having the cell function, that we will discuss in the next class.